Greetings again, BIOL-3 students. Today's lecture will be the first part of Chapter 3, Biological Macromolecules. We're going to be covering 4 categories of macromolecules in this Chapter 3. There are certainly other types of macromolecules or big molecules, but we don't have time in an introductory cell biology class to cover them all. So in this first part, we're going to go through the carbohydrates and the lipids. And in the second part, we will go through the proteins and the nucleic acids. And I believe this chapter will help clarify some of the questions people had about functional groups that were in the Chapter 2 lecture. And our module for this second week also includes some functional group practice worksheets. This image shows lots of different kinds of foods, and like me, you probably don't think about what biological macromolecules are in the foods that you're eating. But this one tray does show us the different types of molecules that we will be discussing in this chapter. So the categories, like I mentioned, are the carbohydrates, the lipids, the proteins, and nucleic acids. And you can think about, for your discussion, which of the foods in this image contain which of those biological macromolecules, and how those relate to the nutrients that we get from those foods. Got a lot of scribbling on here. I'm working on my laptop, and I don't have a way to draw on the slides like I do in the lecture hall on campus, but I will explain these markings as I go through. These hexagons are individual molecules of glucose. We're looking at how biological molecules are built up from their smaller subunits called monomers into larger compounds called polymers. So the suffix mer means a unit, and so a monomer is a single unit, and a polymer is multiple units. And a polymer has to be made of repeating units that have some sort of similar function. In this case, we have 2 individual glucoses. When we join them together, we make maltose. Now, we could combine 2 different individual sugars, like a glucose and a fructose, and we make sucrose. But glucose and fructose are both sugars, so 2 sugars could be combined to make a bigger sugar. We'll see other types of monomers that can be joined to make other molecules. When 2 sugars join, we create what's called a glycosidic bond. That GLYCO, when you see that prefix, means it's a type of sugar or carbohydrate. And the bond, it is covalent because we have solid lines indicating it. And this process that was used to create this glycosidic bond is called dehydration synthesis. Synthesis because we are creating something larger. And dehydration, not because we're drying out the molecules, but because we create water as a byproduct. This glucose molecule has an OH, that is that hydroxy or hydroxyl functional group, and the second one has as well. And when they combine, we take the hydroxyl from one of the glucose molecules and the hydrogen from the hydroxyl on another, and that is what creates the water that is a byproduct. This oxygen wasn't involved in that dehydration synthesis, and it will bind to the carbon, and that is our glycosidic bond. I had to correct the slide. If we start with two molecules that are already joined and we want to break this glycosidic bond, we can do so in the presence of water, and that process is called hydrolysis. Hydro, meaning water, and lysis, L-Y-S-I-S, means to break. So we're not breaking water, but we are using the ionic components of water to break a bond. And it's not spontaneous. If we have a sugar and we stir it in water, it might dissolve, but the individual molecules of that sugar remain covalently bound to each other. We would have to also provide energy and a surface in order to break those covalent bonds. But in terms of a balanced reaction, that would be the case. So we add back the 1H2HO. Those are the ionic components of the water, and we break that bond. We will be talking about enzymes in a later chapter, and enzymes are going to provide a surface where this molecule and the water can sit together and then allow the pressure on those bonds so that we can break apart that covalent bond. As you learned in your chemistry class, covalent bonds are really strong. And because of that, they're not going to spontaneously break apart just in the presence of water. We will also have to provide energy for that. And the naming of the sugars, I apologize that my little box kind of obscures the name. The top one here says glyceraldehyde. The slides are posted without my video face on top of them if you ever need to refer to those in the module. And so these simple sugars are called monosaccharides. So mono is one, saccharide is a sugar. And they get classified based on the position of that carbonyl group. Back to our functional groups, putting them into context now. The carbonyl is a carbon double bound to an oxygen. And if that carbonyl is at the end of a molecule, it is called an aldehyde. And if it is somewhere in the middle of a molecule, it is called a ketone. So an aldose has an aldehyde carbonyl, and so it's at the end of the chain, and a ketose has a ketone. It's in the middle. The way I remembered this is that ketones are like a keyhole in the middle of the molecule, and so aldehyde's the other one. It's on the end. And the ose is a suffix that also indicates that we're dealing with carbohydrates. So a hexose, a 6-carbon sugar, because hex is the prefix that means 6. A pentose is 5 carbons. A triose is 3 carbons. We don't have diose. We have disaccharides, not diose, and you can blame the chemists for that naming system. So the OSE is it's most likely a carbohydrate. Now we combine our previous mentions of isomers with our study of functional groups. So glucose, galactose, and fructose are all isomers. They are structural isomers because they have the exact same chemical formula of C6H12O6, but the atoms are arranged differently. Two different things to note. One of the things that's different is the carbonyl group. It is at the top on both glucose and galactose. So glucose and galactose are aldose sugars. They have an aldehyde carbonyl at the end. Fructose is a ketose sugar. It has a ketone carbonyl carbon. So if both glucose and galactose are aldoses, they have that end carbonyl, the aldehyde carbonyl. If we look through the rest of their 5 carbons, the only difference we see is at carbon 1, 2, 3, 4. There's a hydrogen on the left-hand side in glucose and a hydroxyl on the left-hand side in galactose. And that one little difference makes these 2 different sugars. Remember, this isn't just a drawing. We can't just rotate around that 4th carbon. These are electrons being shared in those valence shells. And so we can't just spin that 4th carbon to reposition the hydrogen and the hydroxyl to be in the same position for both sugars. And that one functional group being in a different position gives it a different structure and means that it will interact differently with other molecules, and in particular with enzymes. Enzymes are made of proteins that we will see in the second half of the chapter 3 lecture. Proteins, when they fold, have a very specific shape, and that very specific shape could detect something as subtle as a hydroxyl being on the left-hand or the right-hand side of a carbon. So we looked at the Fisher projections of those 3 monosaccharides, those 6 carbon sugars, and in solution they tend to form a ring structure. So with glucose, we take that linear Fisher projection and it can form a ring in 2 different formations. We have that sideways fishy is the Greek letter alpha, and this kind of rounded B looking symbol is the Greek letter beta. So we have alpha glucose and beta glucose. So alpha, the hydrogen on carbon 1 is pointing upwards and the hydroxyl downwards. On beta, the hydroxyl is upwards and the hydrogen is downwards. And just like with glucose versus galactose, the left or right position of the hydroxyl group, the up or down position of the hydroxyl group makes these, even though they're glucose, they are isomers that cannot be just overlaid on top of each other. That position makes a difference. We're going to look at some of the polymers that can be built from alpha and or beta glucose, and we'll see that that position makes a difference in how 2 glucose molecules can combine, and once they do combine, the features that that large molecule has. So ribose is a 5 carbon sugar, so is but fructose is a 6 carbon sugar, but its 6th carbon is also sticking up out of the ring. So ribose, each vertex in the ring is a carbon. We have 1, 2, 3, 4, and the 5th carbon sticks out of the ring. Fructose is a 6 carbon molecule, and we have 1, 2, 3, 4 carbons in the ring, but we have 2 carbons out of the ring for a total of 6. Ribose we will see in the second half of this chapter, because ribose and its related molecule deoxyribose are the carbohydrate that is a key component in our nucleic acids. So sucrose is table sugar, the stuff that's in my coffee. I don't really like coffee. I like coffee flavored sweetened coconut milk. So glucose and fructose combine to make sucrose because we are forming a bond between 2 carbohydrates. The name of that bond is a glycosidic bond. We number the carbons by convention based on where the oxygen is in the ring structure. We go clockwise and the first carbon is 1. So in glucose, we have 1, 2, 3, 4, 5. The sixth carbon sticks out of the ring. And in fructose, we're numbering from the opposite direction. They've just rotated over so that we can see where the hydroxyl is. So there's carbon 1, 2, 3, 4, 5, and 6 is sticking out of the ring. Again, two hydroxyls that are in proximity to each other. And so we could take the OH off of one, the H off of another. There is our water released by dehydration synthesis, and we have our glycosidic bond. So disaccharides that we may have heard of, maltose. It's why we add malt during fermentation processes like in beer making. We have lactose, that is the sugar that is in milk, and that is made from one glucose and one galactose. And the bottom one here is sucrose that we saw on the previous slide. So maltose is two alpha glucoses linked together. Sucrose was an alpha glucose and a fructose linked together. And lactose is an alpha glucose and a galactose linked together. But we have two carbohydrates joined by that glycosidic bond in every case. Here we see two different polymers that are forms of starch. We have the top one is amylose, so the OSE tells us that it's a carbohydrate. The bottom one is named amylopectin. Nothing about the name really tells you what it is. These are both, however, forms of starch. And starch is how plants store glucose. They create this polymer of the glucose as a storage molecule. And then when energy is required from that molecule, there are enzymes that can break off and release the individual glucose molecules to go into metabolism. If we look at the cartoons, amylose is this coiled molecule that's more or less linear. And amylopectin has those coils, but then the coils branch off from each other. And that is because of where the glycosidic bond forms between the two molecules of sugar. So we have our linking here. We have two glycosidic bonds shown, and it says that amylose is unbranched chains by an alpha 1,4 glycosidic link. So these are alpha glucoses, so that's why it's an alpha linkage. And we have carbon 1, 2, 3, 4, 5, 6. And here 1, 2, 3, 4. So from the first carbon of one glucose molecule to the fourth carbon of the neighboring one that's being added to the polymer, that is an alpha, because it's alpha glucose, 1, 4. There aren't 14 linkages. It's because of the first carbon and the fourth carbon are being joined in that glycosidic bond. Amylopectin also has those 1, 4 bonds, but it additionally has 1, 6 bonds. That's where the branch is. This coil is all alpha 1,4s. This coil alpha 1,4s, this coil alpha 1,4s. But when we branch, this connection between this coil and that is an alpha 1,6. The connection here is an alpha 1,6. So plants store glucose as starch. Animals, like us and all other animals, store our glucose as glycogen. And it will have similar alpha linkages, but the number of branching is different between them. Cellulose is very linear. Cellulose is a carbohydrate polymer that forms the structure of plant cell walls, and it's also found in some algae cell walls and even in a few other protist cell walls. So cellulose is beta 1,4 bonds. So instead of the alpha glucose we see in starch and glycogen, we have beta glucose. And we see the difference is that we have the individual beta glucose molecules, and because they're beta, in order to get that glycosidic bond between two individual beta glucose molecules, we have to flip them upside down relative to each other. And that's why I circled the oxygens. This beta glucose has the oxygen in its ring pointing downward. This beta glucose has the oxygen in its ring upwards, so down, up. Each beta glucose is upside down relative to its neighbor, and that is how we get the hydroxyls in position next to each other to create the glycosidic bond. And that makes it very linear and much more fibrous, and that thick cell wall that is around all plant cells. Cute little bee is shown here as an example of an insect. Insects are animals, so they have cells like ours that do not have cell walls, but they're here as an example because of their exoskeleton. Right? They do not have an internal skeleton. Their skeleton is outside and crunchy. It's made of chitin, which is a polysaccharide, so it is a carbohydrate. Chitin is also found in fungal cell walls. Fungi are another eukaryote, and they have cell walls, but they are made out of chitin. Now as we move toward lipids, we have a lovely otter here as an example. Otters have many different oils in their fur, and that is what helps them to That's what helps them to repel water, because lipids are hydrophobic, water hating. Keeps them from absorbing water, helps them be more smooth and move through the water more easily, but it also insulates their coats. So this is an example of a triglycerol. It's called a triglycerol. There's only one glycerol molecule. The tri portion comes from the fact that we connect 3 fatty acids. We're going to connect each fatty acid to the glycerol using dehydration synthesis. And I have over here that we can have a saturated fat. It is called saturated because all of the carbon to carbon bonds are single. And it's called saturated because each carbon is saturated or completely surrounded by hydrogens. So each carbon is connected to a carbon by a single bond, and any other bonds around it are to hydrogens. We connect this fatty acid, and I've highlighted here this carbon, double bonded to an oxygen, and single bound to an oxygen that's part of a hydroxyl group here. So this whole square is highlighting the functional group that is the carboxyl group, also called a carboxylic acid functional group. Acidic because when in solution, this portion of it tends to donate the hydrogen ion. And hydrogen ion donors are acids. That's why they're called fatty acids, because they are fat and they have an acidic end. One hydroxyl as part of the carboxylic acid end on the fatty acid, and our glycerol is actually a carbohydrate, and it has 3 hydroxyls. So what the H that's on the glycerol and the OH that's on the fatty acid, that is where we're going to get our dehydration synthesis. And so this is the carboxyl, and this carbon had its oxygen, its hydrogen was donated, this oxygen was still part of the carboxyl group, and that is the bond connecting the fatty acid to the glycerol. So we see here that each carbon is single bound to its neighbor, and each carbon is saturated with hydrogens, and saturated fats tend to be more linear. We've got a little jog in it here, but structurally it is fairly linear because of the single bonds and the saturation of the hydrogens. This is just one example of a triglycerol. In our cells, one triglycerol molecule, that would be this whole thing, the lengths of the carbon chains don't have to be the same. We've got different lengths in each of these, but also the saturation could be different. We could have all saturated, we could have some saturated and some unsaturated. And the unsaturated fats will have one or more carbon-to-carbon double bonds. So saturated fat, this is stearic acid. You won't have to memorize the name of it, but if I showed you a picture, we have our carboxylic acid in, and all of the carbons are saturated or covered with hydrogens. That is a saturated fat. They tend to pack very tightly together, and they also tend to be solid at room temperature. Most examples in biology of saturated fats are animal-derived, but there are some saturated fats that are plant-derived. Like coconut oil, pretty solid, right? Avocados, full of fat, saturated fat, but it's plant-based. Olives, in their solid state, have some saturated fats. In an unsaturated fat, there is at least one carbon-to-carbon double bond, and it's shown here as linear, but it tends to bend the molecule. Because when you have a double bond, you're getting a lot more crowding of those valence shells because you're having to share 2 pairs of electrons. Those shells are going to have to crowd together, and that is going to cause a bend in the molecule. So unsaturated fats have fewer carbons because of the carbon-to-carbon double bond. That makes them more liquid at room temperature. So primarily plant-based fats are unsaturated. And I've circled this here because we don't have hydrogens. Oleic acid and the stearic acid from the previous slide have the same number of carbons, but oleic acid has fewer hydrogens because of the double bond. Carbon can have 4 bonds. We have 1, 2, 3, 4. Around this one, 1, 2, 3, 4. The others are saturated with hydrogens. So this would be a mono unsaturated fat. 1 double bond, we've done 1 unsaturation. If we had a poly unsaturated fat, we would have 2 or more double bonds, meaning many unsaturations. Mentioned cis and trans with regard to structural isomers in the previous lecture. And with regard to fats, it depends on the position of the hydrogens around that double bond. So we have cis-oleic acid and trans-oleic acid. The oleic acid is the unsaturated. So we're looking at, are both of the hydrogens either on the bottom or on the top, or is one hydrogen below and one hydrogen above that double bond. So stearic acid, wholly saturated. Everything is single bonds. Same number of carbons in cis-oleic acid and trans-oleic acid. But with cis-oleic acid, because both hydrogens are on one side of that double bond, that's where we're getting that bend in the molecule because of the crowding and getting both hydrogens on one side. Trans-oleic acid, we have one hydrogen below and one hydrogen above that double bond, and that straightens the molecule out somewhat. When that happens, now it's oleic acid. It has a double bond. It is unsaturated, but because of the different position of those hydrogens, we've created a more linear molecule, and it will act like a saturated fat. You've probably heard the term of trans fats. If you look at a nutrition label, it will tell you the amount of fat that's in that product, and it will list if there are any trans fats. The reason for that is that trans fats are chemically altered to move those hydrogen bonds to make what would have been more liquid at room temperature more solid. It's going to act like a saturated fat. Now, our bodies can certainly process both saturated and unsaturated fats, but our bodies have a tougher time with trans unsaturated fats. This repositioning, same atomic formula, rearranging the atoms will mean our enzymes have a tougher time fitting that. It's supposed to fit this bent molecule, and now it's straight, and it doesn't work as well. Probably have also heard of omega-3 fatty acids. This is alpha-linoleic acid, and I'm not going to make you draw or identify that it is alpha-linoleic acid, but it's an omega-3 fatty acid because of 3 cis double bonds. So we have a double bond here, hydrogens on the same side. A double bond here, hydrogens on the same side, and here. And each time you have the hydrogens on the same side of a double bond, you cause that curve or bend, and when we have 3 of them, we get this sort of looped shape. So that is our omega-3 fatty acid. That's COOH. This oxygen would be double bonded to the carbon, and the OH would also be bound to the carbon. That is our carboxyl or carboxylic acid subgroup. Wax is also a type of lipid. If we look at leaves out in nature, if you pour water on them, they don't just absorb the water through osmosis and get all bloated. They've got their cell walls that protect them, but they also have waxes on them. And so waxes are greatly varied in their structure, but they are all extremely hydrophobic. Phospholipids are incredibly important to our cell membranes. We once again have a glycerol that is here, and we do have 2 fatty acids. A triglycerol would have a 3rd fatty acid attached here, but this is a phospholipid, so instead of a 3rd fatty acid chain, we instead have a phosphate functional group. And so this big circle is drawn around as a cartoon because this is the hydrophilic portion of a phospholipid, meaning it is polar, and it's polar because of that phosphate group. Phosphates have hydroxyl groups, and in solution they donate their hydrogens and become ionized, and that makes them polar. fatty acids are nonpolar. They are hydrophobic. This particular phospholipid is shown as having one fatty acid that is saturated and linear, and it has one fatty acid that has a single double bond and is unsaturated. Just like our triglycerols that could have different lengths and different saturation levels in each of their fatty acids, depending on the organism and even the cell type, the two fatty acids attached to glycerol can be quite different from each other. And certainly between species, there can be dramatic differences in the lengths and the saturation levels of those fatty acids. So here's my very poor drawing of a phosphate. It has on its own 3 hydroxyl groups. We've got the double bond here. We've got one of the OHs there, another one here, and this one has an R on it. It's often just a hydroxyl, but it might have other polar attachments to it, depending on the species. Phospholipids are called amphipathic. Amphi means two, like amphibious, and pathic, pathways. It has both polar and nonpolar regions. This whole thing is one molecule, but part of it is polar and part of it is nonpolar. And that is going to be what allows our cells to use phospholipids in a bilayer, 2 layers, so that we get a double layer of protection, allows for transport, and we'll see how those link together. So all of the phosphate heads are either in the exterior, pointing toward the exterior environment, or toward the interior environment, and all of the hydrophobic tails point inward at each other, because we know that polar things will mix with polar things and nonpolar things will mix with nonpolar things, so there's attraction between the 2 layers at their hydrophobic tails. Steroids are also lipids. Cholesterol is the backbone, sort of the basic structure of all of our steroids. And so we have the cholesterol molecule, and we attach different functional groups to it. This little zigzag means carbon saturated with hydrogen, so this is a saturated hydrocarbon chain. We have ring structures, and the only part of cholesterol that is hydrophilic is this tiny little hydroxyl. It is part of cell membranes in animals, so we need cholesterol in our cell membranes. This tiny little hydroxyl will interact with those polar phosphate heads, and the rest of the molecule interacts with the hydrophobic tails. Cortisol is our stress hormone. You're probably all experiencing that. Try to do things to relax yourself and feel better. So that's one, and you can see it has extremely similar structure. Double bond is moved from here to here, but we have similar functional groups. There is hydrophilic portions in 3 places on cortisol for it to interact with cellular chemicals. We also have testosterone. It's called an anabolic steroid. It is a human hormone, tends to be higher in persons with XY chromosomes and lower in persons with 2X chromosomes, but it's present in all humans. Can be an anabolic steroid. Anabolic means to build up, to make bigger. HGH is human growth hormone, again in all humans. Estrogen, higher levels in XX individuals and lower in XY, but present in all humans. Progesterone is a hormone that helps to maintain pregnancy, and we saw cholesterol, which is part of our cell membranes. So cholesterol is only found in animals. There are no cholesterol in any plant items. There are other sterols, S-T-E-R-O-L, but cholesterol is only in animal products. We will begin the next lecture here with the amino acids and get into proteins. So the first lecture of week 2, you're responsible for the carbohydrates through the lipids for your discussion.